And I have some like what I think is globally like exciting news. The Nobel Prize for Chemistry was announced this morning. And did anyone happen to see who was one of the recipients of it? There are a total of three people that it was distributed to, or that will be sharing it for chemistry. Um, but one of them is a fellow by the name of David Baker. And does anyone, if you didn't see who won it, did you happen to see what they're kind of recognized for? Specifically David Baker. But they kind of share it for the same topic. Not RNA, but what David Baker is kind of known for is protein folding and predicting protein structures. So David Baker has been at the University of Washington since I would say like the mid 90s or so. And what his research group studies is basically trying to use computational techniques to predict the structure of a protein, the 3D structure, that tertiary structure of a protein. So I thought that was like particularly timely or prescient for what we're talking about in our class. So that's pretty exciting and pretty awesome because he is one of a handful of people who just drove that field forward tremendously. His research group has like 60 some odd people in it. It's yeah, uh, it's almost like an entity unto itself because he's like the big boss, but then he's got several research scientists underneath him, and yeah, so the hierarchy is kind of interesting, and it brings up the hierarchy of research and how it's conducted and whatnot. So this is where we took off. We had this forces stabilizing tertiary structure, which we're going to look at that, and then we had a question that followed this. We're going to skip over the question because we did it last time. But these were, I'm going to kill my video just because I want to make sure that I just got a notification that my internet was funky. So I've stopped my video. Forces stabilizing tertiary structure. We have hydrogen bonds, disulfide bonds, hydrogen bonds which stabilize the secondary structure. But there are hydrogen bonds that stabilize the tertiary structure as well. The difference is that the hydrogen bonds that stabilize the tertiary structure would be hydrogen bond interactions between R groups and R group backbone interactions. Hydrogen bonds that stabilize backbone interactions are exclusively for the secondary structure. There's hydrophobic interactions, ionic bonds, and metallic bonds as well. Now, to move forward just a tad. So proteins fold into this 3D conformation, and that 3D conformation is one that is kind of its native state, its best state. Um, Whenever we study proteins, one of the things that we always want to do is be able to analyze them as basically their primary structure and be able to therefore denature or unfold them. Now, in order to unfold or denature a protein, you need to be able to break any of those forces that stabilize the tertiary structure. So, as an example of those, when we talk about a disulfide bond, does anyone remember the amino acid or the amino acids that form disulfide bonds? And whenever I say acids, I mean there are two amino acids. Sorry, two amino acids are essential for this, but there's only one amino acid that does this. Does anyone remember what that amino acid is? Close, close. It's not methionine. That also has a sulfur, but cysteine. So CYS is our three-letter code. So cysteine and cysteine, they will form a disulfide bond. Andrew, that's a great point to bring up of methionine. But for that to happen, it's not simply an ionization. Instead, you'd have to break a covalent bond between that carbon, because if you remember, methionine is basically CH2, CH3. So this amino acid would have to be demethylated in order for that to happen. So disulfide bonds are going to form between cysteine-cysteine residues. Now, there is a reagent, or there's two different major reagents that break those disulfide bonds. They're known as mercaptoethanol or dithiothriotol. Now, this mercaptoethanol, a little bit more precision on its name, it's beta mercaptoethanol, or what sometimes people refer to it as is BME, or you might sometimes also see 2 mercaptoethanol or 2-ME. Now, dithiothriotol is DTT. And when it comes to these two different reagents, why one over the other? There's not, it's almost just a preference. They're both equally cheap. I would say that they're both equally abundant in unfolding proteins. But what I want to bring up is that this reagent is essential if you have a protein that has any disulfide bonds in order to break that bond. If you don't have or if you have a protein that doesn't have any disulfide bonds, you arguably don't have to worry about this. Now, that's really a question for when you know your protein a little bit better. Now, that's one of the forces or one of the reagents or one of the different reagents that can break a stabilizing force of the tertiary structure. The others are a little bit more general, and I'm going to talk about them all kind of collectively. So, all of these reagents are pretty much used whenever you're doing any sort of denaturation studies. So, to unfold a protein, you're going to heat it in the presence of a detergent, also in the presence of urea and guanidinium hydrochloride. Urea and guanidinium hydrochloride, you might see written as GNHCl or GDHCl. Now, what guanidinium hydrochloride and urea do is they are known as hygroscopic agents. And what hygroscopic agents do is they disrupt hydrogen bonds, which is stated right here. So basically, what they're going to do is destroy any sort of secondary structure because secondary structure is going to be stabilized by hydrogen bonds. But also they'll disrupt hydrogen bonds between R groups. Now, in addition to using urea and guanidinium hydrochloride, what's also pretty common to do is adjust the pH. So change the pH of the solution. Because what that will do is disrupt any sort of charge-charge interactions or ionic interactions. Sorry, and any sort of ionic interactions. Now, a detergent that's commonly used is known as SDS. Now, that's not the only detergent, but SDS is one of the common reagents that's used because if you've ever had the opportunity to run a protein gel, that is an SDS page. Absolutely. So SDS page. Now, this is like a $5 question, but does anyone know what SDS stands for? And I know that's a big ask because it's kind of a long word or kind of long words, but I suppose if you don't want to type it all out, you could just give like a thumbs up or something to the chat. So SDS is it's a detergent, and what it does, or what it's known as, is sodium. Yes, absolutely right, Morgan. Sodium dodecyl sulfate. Now, when you see that dodecyl, and actually when you see that DEC prefix, that's going to indicate 10. Dodecyl would be 2. So this is a compound that has 12 carbons. A 12 carbon compound plus sulfate plus a sodium ion. Now, the role of SDS, what it is used for is, or the kind of intent or purpose of it is it gives proteins or a protein or a polypeptide a uniform negative charge. Now, this is for an application that we'll talk about in a couple of weeks, but I want you to have this understanding because when you think about a protein in an aqueous environment, well, that protein can have positively charged residues, negatively charged residues, uncharged residues. When you put or when you incubate your protein with SDS, what happens is, let's say here's our linear protein. What happens is I'm going to draw a little coating basically. Maybe I should have changed colors there because now it just kind of looks like a little worm or something like that. But basically, it gives the protein negative charges all along its surface. And the number of negative charges corresponds to the number of amino acids. So when you think about what SDS is doing, well, it's helping to denature because it's going to disrupt hydrophobic interactions and basically give them a negative charge. Similarly, it's going to take any sort of positively charged species and give them a negative charge. And any negatively charged species, it's going to basically replace that negative charge with another negative charge. So the intention here is to take basically this wonderfully folded 3D structure, which I'm going to go back to right, actually check right here. This folded structure where there's some organization and put it into this kind of linear or semi-linear, it almost looks like a shoestring of sorts. And SDS is one of the reagents that does that. So whenever you're unfolding a protein or denaturing a protein, you're going to use all of these different reagents in order to do that because you have so many different reactants or so many different forces that are stabilizing that structure. Now, in addition to that, the last one that you might see from time to time is a chelator. And a chelator is a class of compounds. And actually, if there's anyone who's ever heard of, well, I'll ask kind of a couple of questions. First of all, has anyone ever heard of chelation? And one way that you've possibly heard about it is in a clinical setting, chelation therapy. So awesome. So chelation therapy is a technique that's used to whenever someone has like heavy metal poisoning, or maybe if someone has, one way that it can possibly happen is by basically um, if you were to get like an iron supplement or something like that and just eat the whole bottle, well, then what's going to happen is you got a lot of iron, excess iron in your body, and that can cause damage to your liver and your heart, lots of different muscles. In which case you would use a chelator. One in particular is EDTA. Now, I don't need you to know that chelator. I just want to bring that one up as a common chelator. Um, And what that does is it basically finds a metal ion like magnesium 2 plus. And basically it almost like encapsulates it in some respects so that that metal ion is not going to participate in any sort of um, metallic bond like what we see here with our iron ion. So that's one of the ways that you can disrupt that interaction there with a chelator. So these are all different um, reagents that are regularly used to denature and unfold proteins. Now, I'm going to clear that. Now, this is a little bit, so you got to look at this kind of on its side, but also kind of straight up. So this right here is a model for a classical experiment. Now, this was the enzyme RNase. This enzyme was purified in the I think it was like 1948 or so. I'm going to say 1940s to 50s. And this enzyme was, purified and it it served as a great model for, or it served as a model for characterization of proteins and protein structure studies. So here what we have at the top is our folded ribonuclease or our folded RNase. Now, what this enzyme does is it sees an RNA molecule and just kind of chomps it up. It takes that RNA molecule and breaks it down into individual ribonucleotides. At the bottom, what we have is our denatured or our fully denatured form of RNase. So there was a fellow by the name of Christian Anfinsen. Christian Anfinsen. And what he did was he said, we can unfold this protein. I'm curious if whenever we add a bunch of urea and beta mercaptoethanol, so effectively when we break up our hydrogen bonds and then we add the BME to break our disulfide bonds, our cis-cis residues, or our cis-cis double bonds, like, yeah, we're good. We know that we can unfold it. But what I'm curious about is what about if we take a denatured and unfolded form of that protein and we remove the urea? So we basically titrate it away. So remove the urea and remove the beta mercaptoethanol. What's going to happen to my protein? And what Christian Anfinsen found was that you could refold an unfolded protein. Now, that was like it, the significance of this was that essentially it meant that the primary structure gave the protein all of the information. So I'm going to say gave all info or all needed to form the tertiary structure. So this is almost like, okay, cool. If we can take a functioning protein and break it and then fix it, then essentially everything that is needed for that protein to become functional is available in the primary structure. Now, this is huge because of the fact that it it kind of gave someone like David Baker, current Nobel recipient or Nobel laureate, a platform to stand on. And of course, this is back in the 1950s, probably when David Baker was like two. But this is like the work that was done by a giant that we can now stand on their shoulders. So this primary structure gives way to, there's not some magical mysticism taking place in a cell that gets a protein to take on its folded 3D structure. Instead, all that you need for that 3D structure is the primary structure of the protein. So essentially, it was a way or another, a roundabout way of saying is the primary structure enables us as kind of scientists to, or us the ability to predict the tertiary structure of a protein. So this is awesome because then all of a sudden it's like, oh, structural biology is a fascinating field. Structural biology is a worthwhile field because then we can study, we can reduce and process this information. Knowing the primary structure is important because if you know the primary structure, well, then maybe you'd be able to predict the secondary structure. And if you can predict the secondary structure, then you can predict the tertiary structure. You can predict these higher levels based on a, a fraction of the information. So that's what I want you to know about Christian Anfinsen is his kind of impact on structural biology. He studied, so that the takeaway of this is that Christian Anfinsen studied the folding and unfolding of RNase A or RNase. We'll just go with RNase. And that served as a model to basically be able to take the tertiary structure of, or sorry, the primary structure of a protein and predict the tertiary structure of a protein. So this was a big deal to say the least. Okay. Now, a globular protein going from that unfolded to the folded state and the process of getting from an unfolded protein to a folded protein is a huge deal. It's like a big question. It's almost like a philosophical and an abstract question. Now, what is shown here is what's known as a folding funnel. A folding funnel and it basically says that this right here where I've drawn an X and now where I'm drawing a Y, these are two different unfolded forms of a protein, the exact same protein. So the shape and the kind of like yeah, the shape of the unfolded protein there can be multiple different shapes of that unfolded protein. They will, however, have one kind of agreed upon functional native state, and that's going to be depicted down here at the bottom of this. What this is showing is that these two different unfolded proteins, these are high energy molecules. They are very unstable, and so that means they're going to be moving all over the place. What they will do is they will undergo conformational or shape changes until they reach their native state or their folded state. Now, I have an X and a Y here because these two different unfolded proteins are going to take two different paths to get to the same place. Now, what this also means is that your native state is your lowest energy state of a protein. So native state equals lowest E state of protein. It's also your most stable, in part because it is the lowest energy state. It's not like fighting to change or anything of that nature. Now, these folding funnels can actually be much more complicated because of the fact that it's not simply two different shapes. It's not an X and a Y. Instead, there are thousands of different permutations that a polypeptide is going to start at in its unfolded state. And so whenever I say a folding funnel, really what this is if you have a top-down view of it, there's tons of different paths here, 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 here. What I want you to know about this is that the unfolded state of a protein is the high energy state. It's going to go through a number of different reorganizations and movements where it will go through a partially folded or a molten globule shape until it reaches its high energy or its highest energy, sorry, its lowest energy native folded state. Now, when it comes to how a protein folds and what this process actually kind of looks like, there are different parts of a protein that will fold in different rates. And we're not going to talk about rates. That's more of a biophysics class sort of question. So if you are interested in this and want to go on to graduate school, you'd take like a biophysics class where you learn about, well, calculating free energies of protein structures. But proteins have different domains. So an example of that is when you look at a protein and it's, 700 amino acids. So let's say that we have a protein that is, 1 to amino acid 700. There is a portion of that protein, which I'm going to shade in red here, that has a particular function. in addition to its particular function, it's also going to adopt a specific shape. Proteins have structural domains and functional domains where the overlap between the structure and the function are very similar. So what that means is that if you and they have patterns. So if we go ahead, jog ahead for just a second. Um, if you were to take the primary structure of a protein and do a secondary structure prediction, if you were to predict the secondary structures that form, what you might find is a pattern. Well, here's a helix followed by a turn followed by a helix. Or you might have a helix followed by a random loop or a coil followed by another helix. Now, as you study proteins and their structures more and more, you might notice that, oh, well, whenever I see this helix turn helix, someone's already characterized a protein with that, and that has the ability to bind DNA. So this structure right here, this helix turn helix structure, well, that will be characterized as a DNA binding domain. So anytime you see helix turn helix as an example, you would then say, oh, well, I bet this protein has the ability to bind DNA. Now, or yeah, so this is one of the things that biochemists and structural biologists study is they look at, well, what are the structures that we see from the primary structure? What do we see in the secondary structure? And where have we seen that secondary structure before? Because that gives us a mechanism to predict what this protein is going to do and how it's going to function. So there's a number of different structural domains. And what I want you to know about this is that really this is kind of a concept, but structure of a protein, secondary structure of a protein gives you kind of information to predict function. And I want you to kind of accept and understand as a concept that there is a strong relationship between the function and the structure of a protein. I don't want you to keep these in mind of like, oh, protein, a leucine zipper will be mediating protein DNA interactions or protein protein interactions. You'll get into those if you take a molecular biology class where you'll talk about, well, this domain does this. But I do want you to understand the relationship between structure and function. Now, after our tertiary structure of proteins, we get into what's known as the secondary structure is a way that you can predict because secondary structure is going to say, oh, well, there's an alpha helix here, there's a beta sheet here, there's an alpha helix here. Your tertiary structure is where you're kind of like putting that all together because simply from the secondary structure, you don't know if you can look at the sequence and say, okay, well, there's an alpha helix here, but you don't actually, and there's an alpha helix here, but you don't actually know that those alpha helices come together. So secondary structure can be kind of like a suggestive, but it's the tertiary structure that's more conclusive. So, quaternary structure of a protein does that answer your question, Andrew? Okay, great. So quaternary structure is our highest level of organization of a protein. Now, this is where proteins kind of aggregate together, where multiple polypeptides fold together. Tertiary structure is one protein and how it folds independently. Quaternary structure is where multiple polypeptides fold together. Now, these polypeptides are referred to as subunits, and they are different polypeptide chains. So the individual subunits associate in a specific shape known as the quaternary structure. Now, subunits interact through non-covalent interactions. That's an important point to keep in mind that this is for globular proteins. So globular quaternary structure proteins interact through non-covalent interactions. Now, proteins with more than one subunit are known as oligomers or oligomers, a dimer, trimer, tetramer, pentamer, hexamer, heptamer, octamer, nonamer, decamer. Can't remember what 11 is, but 12 would be a dodecamer. Um, so when you have multiple subunits kind of aggregate together or multiple polypeptides aggregate together and form one complete thing, that would be an oligomer. Now, so the ribosomes, yes, they are. The ribosome is like a superstructure, though, because um, those subunits fold together, but then the ribosome has other components as well. So um, I would say in the simplest answer, yes. But then it forms something larger than any like, you know, multi-protein structure. So it's yeah. A protein with five identical subunits is best known as a. We've got 1 minute on this questioner. So one important part is that these are identical subunits. All right. So we were looking for a homopentamer. Pretty simple question there. So I'm happy to see that response. Now, when it comes to, so if they weren't identical, absolutely. And we're going to get into a hetero oligomer in just a minute. Um, Now, in terms of proteins and their sizes, here we've got a protein that is as small as 30 amino acids. Proteinase inhibitor 3. This is 30 amino acids. It's approximately 3,427 Daltons. One Dalton, or sorry, the average one amino acid is approximately 110 Daltons. Now, a Dalton is probably a unit that you haven't heard, or if you have, it's maybe been a minute. One Dalton is equal to 1 gram per mole. So the average amino acid, and this is kind of a convention, regardless of what that amino acid, whether it's glycine or arginine, we treat it as 110 Daltons or 0.110 kilodaltons. Um, but then the largest protein is a structural protein known as titin. Every time I see that word, I always want to say Tintin. I don't know if anyone read that comic book when they were a kid, but anyway, titin is a protein that's 34,350 amino acids, so it has a molecular weight of 3.8 million Daltons. So it's pretty massive. In addition to that, what's significant is that it is a single subunit. Now, there's two proteins on here that I want to highlight in addition to that, myoglobin and hemoglobin. Myoglobin is a monomer. Hemoglobin is a tetramer. They are 153 and 574 Daltons, respectively. Now I'm going to pause my share because that's where we're going to go next. We're going to talk about the function of, oopsies, the function of the protein hemoglobin. Now, there we go, and resume. So, protein function. What I want you to first recognize is that we're using these proteins or the proteins that we're about to talk about, we're going to be using them as a model. And the first thing is that we want to talk about these proteins as a model for how a protein has the function to bind something. So protein ligand binding. Some people pronounce that word ligand, and I don't know that there's a right or a wrong. I say ligand. Maybe that's like a classist differentiation. I'm not sure. But a protein and ligand interaction. Now, essentially, what we're talking about is one protein's ability to, my hand being the protein, bind something. Now, that binding, one of the important parts about this is that this is not catalytic. So essentially what that means is that my right hand binds this timer cube and releases it. My hand doesn't change after the release and then the cube that I bound also does not change. So the function of many proteins is that they simply bind something, hold on to it, and release it. They're not catalyzing a reaction necessarily. Now, those ligands, I use this little timer cube, they can be a variety of different things. They could be, they could be six carbon molecules. They could be inorganic ions. Excuse me. They could be inorganic compounds. They could also be proteins. So ligands can be a number of different things. But protein ligand binding is not catalytic. It regularly causes the protein, my hand as the protein, to undergo a conformational or a shape change. Now, this shape change was very minor. My hand just kind of clenched down on it. And that would be similar to what the protein does, a very minor change to the structure. This is quite often a regulatory step or regulatory in nature. Now, to illustrate this, like I said, we're going to use hemoglobin and myoglobin as our reaction or as our models. Now, what hemoglobin is going to do is it is a protein that will, in the lungs, pick up oxygen. Hemoglobin can transport a total of 1, 2, 3, 4 O2 molecules through the arteries into the tissue where it deposits and releases O2. And then hemoglobin will continue. Hemoglobin can pick up CO2 and it will transport that CO2 as HCO3 and then release it in the lungs as CO2. CO2 is insoluble, or is CO2 and O2 are fairly insoluble. So essentially that hemoglobin is helping to solubilize these. Now there's a couple of other things involved in this right here, but that's not for today. So this O2 that gets deposited in the tissues, it has basically two different routes. One, it can go into the mitochondria, which is also known as that's right, the powerhouse of the cell. I'm guessing at least one person sitting in their own home did this, set it to themselves, and then there's probably someone else who's ready to type it in. But the mitochondria is the powerhouse of the cell, and it requires O2 to produce ATP. Now, that O2 that is deposited by hemoglobin, it could also go to myoglobin, where myoglobin is going to hold on to that. And then when the time is right and whenever that O2 is needed, that myoglobin will release O2 so that it can go into the mitochondria. So essentially, hemoglobin traffics oxygen from a high O2 concentration environment to a low O2 concentration environment so that it can be used out there. Now, that's that's the name of the game for hemoglobin. Now, that's a great question, Andrew. Yes, 4O2 in and the number of CO2 out, it's a little bit variable because of the fact that hemoglobin can bind CO2 in potentially 8 different places. And those 8 different places are on the N termini of all the different subunits. Um, but I think that if you just go with, and then it can also, it has a loose binding at the iron binding site. Um, I think I would modify that and I would just say 4 in and actually no, let's keep it at that because that you have a good reference point of um, the two CO2s out. So yeah, we'll go 4O2 in and two CO2 out. And the only reason I'm a little bit iffy and wishy-washy on my response is because of the fact that there's a what can like whenever hemoglobin deposits oxygen, it doesn't always completely unload all 4 oxygens. It's only going to really hemoglobin because we start talking about hemoglobin as a population of thousands of copies of hemoglobin, and really about 30 to 40% of the oxygen that is carried to the cells or to the tissues gets deposited. But we'll keep it at that 4 in and 2 out. I like that model. We'll go with that. But in the future, we'll kind of put a little bit of a wrinkle to that. Okay, so hemoglobin and myoglobin, we talk about them as models for protein function, because they're, it's close to our heart. It's essential. Myoglobin functions as an oxygen storage and transport protein in the muscles, whereas hemoglobin functions in the same way, but in blood vessels. Now, in addition to those two proteins having those functions, structurally, they're very similar to one another. If we look at the model on the left-hand side here, we've got myoglobin or MB, and I'll use that MB and HB to denote hemoglobin and myoglobin. Now, myoglobin and hemoglobin are built on what's called a structural motif known as a globin fold. Now, the peptide chains fold around something known as a prosthetic group, known as heme. Just like that prosthetic group that was a part, or when we talked about prosthetic groups the other day as being a part of the tertiary structure, they can also be a part of a quaternary structure. Here's our heme molecule. Now, what I always like to point out whenever we look at the structure of myoglobin and later for hemoglobin, when you see these letters A, B, C, D, E, F, G, and then in the background here H, those are all alpha helices. So hemoglobin and myoglobin, or a myoglobin protein, is just a series of alpha helices that have folded into this kind of unique shape known as a globin fold. So this is a globin fold made up of a bunch of alpha helices. Now, if we look at hemoglobin, we've got the same A, B, C, and so on. That same globin fold. Now, one thing that's important to recognize about hemoglobin versus myoglobin is myoglobin and hemoglobin are not basically the same protein. So hemoglobin, HB, is not for myoglobins. The primary sequence of these proteins are going to be a little bit different. So they are distinct from one another, or they're different enough to be distinct proteins. Now, hemoglobin is what's known as a dimer of dimers. If you notice down here, we've got alpha 1, alpha 2, beta 2, and then back here is beta 1. So this is a dimer of dimers. There's two alpha beta dimers that come together to give us the overall structure of of hemoglobin, I'm sorry. Now, these proteins are going to fold around this prosthetic group, this heme molecule. And we'll get into the structure after this next exam. We're going to get more into the structure of hemoglobin and its dynamics. But here we are right now. Now, here's a zoomed in view of myoglobin. Myoglobin is a largely spherical protein, and about 90% of it is alpha helices. Same thing goes with hemoglobin subunits. They're roughly 90% alpha helix. That's not to say that those proteins are the same. Now, one thing that's worth recognizing here is where we saw A, B, C, D. Now you see A, B, C, D, F, G. That is simply referencing the turns that exist between these alpha helices. So in terms of if you were to look at that CD right here, or that AB right here, or FG, you have a kind of abrupt turn. Now, if you were to predict what amino acid was in one of those locations, chances are it'd be good to guess something that's either rigid, like a proline, or something that's very flexible, like a glycine. Now, that's just sort of a thing that I want you to be able to like, critically think about. Um, so then if we look a little bit further or closer at our heme molecule, our heme ring. Heme is what's known as a porphyrin ring, and it has an Fe2 plus ion at the center of it. Now, one thing with respect to Fe2 plus is that this has to be Fe2 plus in the center of that ring. If it's Fe3 plus, its ability to bind O2 just plummets. Now, the what's worth noting about this is that this iron ion is basically in a flat plane with these four different rings here. Now, the amino acid that's down below that is going to be the same amino acid that is above it. So, okay, here is my flat plane. Here is the histidine that's below that helps coordinate and position the iron ion that basically binds the O2, and then there's another histidine above it that helps coordinate and position O2 above that iron ion. Now, as a little bit of a question here, what amino acid positions the iron ion at the center of a heme ring?
Oh, Lord. I'm about to do my meeting.
So what we're going to do is after everyone's come up with an answer for this question, we got our answer of histidine. Cytosine is not an amino acid. Um, when you think about this sort of question, one of the things that I want you to think about is what type of, or this amino acid is an ionizable amino acid. To basically attract an iron ion, you got to have some lone pairs of electrons for that iron ion to want those electrons to kind of be coordinated by them. I got a private message of, is Friday review or finishing this chapter? Well, it is going to be the last few slides of this presentation, and then we are going to talk about the exam. But the most popular answer here, histidine, is the correct answer. I think what I'll do is I'll just go ahead and stop it right there. We've got basically four more slides. We're going to talk about something known as allostery on Friday, and then we'll go from there. Now, Before I part, though, I've got two quizzes that I posted, or two homework quiz things that I posted on Canvas that's got some good questions that you should kind of or that you should answer and look at in advance of your exam on Monday, or our exam on Monday. Um, I will have the exam done by Friday, so I'll have something to, or I'll have more information for you guys then. But it's in the works. I'm using the learning objectives that I gave you. Um, and if you have any questions, if you want to meet with me, please let me know. I'd love to meet with you today or Friday. Just let me know. Other than that, I hope you have a wonderful day, and I will see you guys on Friday. They are graded, so they are mandatory. What time will the exam open on Monday? The exam opens on Monday at, I believe it is, our class starts at 9, so it'll open at 8:30. All right, thank you guys.